Okay, so we're going to talk about alignments and some aligners. So as everybody for now, we've done the quality control part of the data. You kind of understand that. Most of you sent me your homeworks and emails, and that's fine. It was great. There weren't any. They look almost alike because the samples are from the same run, and we only took 1,000 lines. So that's why the files are short, but that's why they look identical. If you were to sequence them throughout, not sequence, like take the entire file, it would probably have some differences. And some of you may have noticed that the reason the files have the first few bases like that, that's not important, but the fact that we can cut that off, that's the important observation to make. There are tools like FastX Clipper from the HANA lab, which you can use to chop off some of those first few sequences. Could be adapter contamination or whatever it is. And FastX adapter search also helps you come up with, what are the possible contaminants that have occurred. So, anyways, so the next step after we're done with the quality control is to align our file to the genome, and you can have any genome or any ohm, transcriptome. You can pretty much choose your reference alignment based on what you're working on. And the three things that you need for that is the FASTQ file, which you will have from your the reference GTF. So this is the part, the GTF format that we covered last time. The GFF and the GTF part we were talking about. This contains the units of your file. So like, where does a exon begin? Where does a coding sequence begin? All those things. And then there's the FASTA file, which is basically your genome FASTA. So it has all the chromosome and all that information. If you want, we can quickly look into the UCSC Genome Browser. I don't think you all need to do this. I'm just going to quickly pull up have you used the UCSC Genome Browser? Has anybody used it?
At some point in a genetics class.
Yeah, 86.
Okay. Right, so if you go here, so you have the Genome Browser, you have the Cancer Browser as well. We won't be covering this, but this also has some useful data sets here. So, okay, so this is your Genome Browser, right? It's hard to fit everything. Okay, so the way you can download stuff is, you can choose your group based on, normally we work with mammals, so human, mouse, whichever organism you're working on. And then this is an important one. So the assembly, most people have switched to this latest HG38 assembly, but there are still some people who use HG19 for, depending on what your data is, or your previous alignments have been made with HG19. So it's very important. These reference alignments are very different. So HG19 and 38 are very different. There's a lot of starting of your genes will be all different based on, so you have to make sure you choose the correct one, especially if you're comparing across samples that were aligned previously by some other person. It's important to find out what your reference alignment was and use the same one, or take those FASTQ files and realign them to the new reference. Okay? Alright, so then after that, I think there is one. So here you can enter your position. So this will basically give you, what it looks like. But for us, so like, you can go into details here, see this is the position of interest, blah, blah, blah. But for us, the more important one tool that we use is under Tools, if you go to Table Browser. So this is the part where you can actually download sequences, right? So this is where what I use to download the GTF file that I was talking about. You can download from here. So let's say these three fields stay the same, but then here is where you can choose whether it's gene and gene prediction, you just want the mRNA or something else, you know, depending on the expression, regulation, all these tracks, they're different. And then within that, you can see whether you want the ref-seq genes or the link RNA, RNA-seq data, or, you know, anything else. Let's say ref-seq. And then within this, you can choose the table type, like is it ref gene or is it, you know, ref flat. I usually end up choosing ref flat.
What is ref flat?
It's a format, so the way the data will be in. I mean, you could also, you know, you could just go with, let's say we choose gen code here. So then see how this thing changes based on that.
Yeah.
So you could, you know, choose whichever other variation you want from this.
Sorry, what is gen code?
It's basically, again, it's like these are all different types of tracks. So like ref-seq is their own version of genome tracks that they have. They did their own genome assembly in one particular way. So these genes will be different. The IDs are different. So UCSC also has their UCSC IDs. So that's part of bioinformatics, is, you know, kind to know what your reference is. So you can also go to Ensemble and download your reference genome from there. So that will have Ensemble IDs instead of, so there's known genes, then there is ref-seq gene IDs, and then there's UCSC gene IDs, and then there's Ensemble gene IDs. So these are all different, you know, like.
So sorry, are these a genome assembly, like the entire
Yes.
genome, or is this just the
The way the
transcriptome?
genome, no, this is the way the genome has been divided exactly
Presented,
into,
yeah.
exactly, it's presented into their specific IDs. So like the genome
okay.
sequence is defined as a consensus. That is HG38 that you have downloaded the FASTA file, it's already there on our server. But then the next step is how do you quantify into units? Like, you know, where is the coding sequence, where
so
is
just
the
like
ID,
Okay, cool.
which ID do you define this as? So do you consider, you know, like those are the things, like basically how do you present the genome? That's a
Okay.
good way to put it. So anyways, so,
Like if you go to PubMed, there's different versions
Right.
of the information you can get and different IDs associated with it. It's just different, yeah, depending on what you like to use.
Okay, cool. Thanks.
so anyway, so let's say, you know, you say this, you want to just look at all the human mRNAs, I'm just saying. So you can get output here. Now, one way is you can, you know, get the output like this on your browser, but if you wanted to save it to a file, you can save this output via specifying an output file name here. Okay? So just a quick way to say that, you know, we can download this data in multiple formats, and then if you want, we can cover more of this in detail, or, you know, if you feel like downloading stuff, I can help you out. But most of the files that you need are there on the server. Okay? So you will not have to probably download stuff. You can just use the UCSC Genome Browser to view the results and the tracks that you come up with, as opposed to downloading these reference files and stuff. So they are, I'm almost 100% sure they're already there on the server. Okay? All right. So, just wanted to quickly go over this. Okay.
So if you wanted to, just a quick question on Iris one, if you wanted to put your own, let's say you have a model organism that's not used, that's not on Iris one, can you actually upload that?
Yeah,
and keep that?
sure.
Does that become publicly available, or is that something?
Not necessarily. If you
just
put
keep
it in the
your-
public folders, then it is. So remember the permissions?
Yeah, yeah.
So if you put it just for your lab, then you can have your own version.
Okay.
In fact, a lot of people end up duplicating the human genome because they will not use the reference one that is there. They can create your own reference, but even that is okay as long as your entire lab is not creating separate versions of the same thing.
Sure.
You know, so like if I'm part of one lab, then I can use that one. But if I'm creating multiple versions for my different projects and different, then it gets really just redundant amount of data. Okay, so then the next step is the aligners. So we have these alignment softwares. So like there's Tophat suite, there's Novo Align, there's Star Aligner. And then there, over here on this link, so if you go on this link, you'll be able to find that all the different aligners and all the different alignment algorithms, right? So feel free to visit this for your own intellectual curiosity. You don't need to, but if you are curious, that's definitely one option. So different aligners use different algorithms. So Bowtie has, it's part of the Tophat suite. So this is by Cole Trapnell, and who is now, I think he's at Harvard now. He was at WashU. So Bowtie is an aligner. It aligns short DNA sequences reads to the human genome at 25 million reads per hour on a typical workstation with 2 GB of memory. So now the main thing which I don't want to go into detail is these Burrows-Wheeler transforms or indexes. To give you a very general idea of how this is done, see, human genome is really large. So if you think of it as a book, it would be like this really almost, the Encyclopedia Britannica, for example. Think of all of the volumes of that Britannica combined into one book. That would be your human genome. Now, you come up with a sentence, and your task is to find that sentence in this volume of books. So that is a very tough problem. So computer science people have worked on this problem for many, many decades, and they're trying to find fast ways to, what is the fastest way to find this exact sequence of characters in this big volume of data, right? So one way to do it, and this has turned out to be the most efficient and the fastest, is to actually create what we call as an index, which is very similar to your book index. So if you have that Encyclopedia Britannica, you go to the back, it tells you, right, so it's starting alphabetically, this is the subject related to what you're looking at, and then you go reference that index, and it directly takes you to a page number where you have information about that topic. Does that make sense? So computer science people also do that. So there are multiple ways. You can create, these index for the entire human genome. So it tells you a little bit about the sequence, start and say that, all sequences that look like this, where are they located in the genome, So those are the ways to create, the way to create an index is using these different transform, like Burrows-Wheeler's transform, where you transform, why it's called a transform is because you take the entire genome and then you transform it into an index. And there is a lot of mathematics behind that on how you do it. I will not be covering that in this course for sure. I don't know if you want to go over it, I can go over with you specifically, but then there's also something like, just like the Burrows-Wheeler's transform, Star Aligner uses something called a Suffix Trees. So Suffix Trees, I'm sorry, they're very memory intensive. They take, for example, like if this is a suffix tree is a sentence, then they create a graph out of each character so that, it looks like a tree where, so if you wanted to search for THI, it'll be like the first node, and then THIS could go into one branch, and then is goes into the next branch like that. Think of it as a lexical graph of characters. Now imagine the advantage of that is, like, for any combination of letters, you will have that instantly. So if you that's the way the tree is constructed, that instantly you, within a fraction of a second, where and how the sequence exists. All right. So that's the main advantage, that basically in a unit of time, like a fraction of a second, 1 unit of time, you're able to find that out. Again, suffix trees are a whole different, there are papers and pages of information on suffix trees. So we're not going to cover that, but I just wanted you to know that these are fast, efficient ways. Now, Burrows-Wheeler transform is not as fast as suffix trees, but suffix trees, because you're trying to create an alphabetical graph of the entire genome, they take up a huge amount of space. So every index will take some extra space, and that index is loaded into the memory when your aligner is aligning. And by memory, I mean the RAM of the server. So for example, when bowtie aligns your sequence, it loads this index into the memory, and that's how it references all your FASTQ file sequence reads and finds out, okay, this read matches in this location, let me go put it there. Or this read partially matches this location, and then let me go there and see how many mismatches are allowed, and how can I proceed from there, right? Now, Porter's-Wheeler transform is based on an algorithm that is, approximate. So there are approximate algorithms and there are exact algorithms, which, as the name suggests, exact algorithms will be able to find you the full sequence read match instantly, and approximate algorithms like BLAST, they only do a small sequence search, and then they expand on both sides of that sequence. So they'll take a small k-mer, or by k-mer, k is the number, and mer is like, so 5-mer, 2-mer, 3-mer. So they'll take a small k-mer unit and look for that sequence from match in the genome, and once it finds multiple locations, it'll search each exact location and expand in both sides and see how accurate that match is at multiple locations. And then if it turns out one location is better than the other in terms of match, it'll pick that as the first preference, or depending on the user preferences, whether you want multiple matches. If there's exact matches in multiple locations, you can have the option to either ignore all multiple matches, or you can have specify that you want them to be reported and count the read twice. So your read will be aligned twice in those locations, right? But those are all things that are covered. So if some of you went to read the manual, so there is this option where you can specify what to do in this kind of situation in your Tophat or in the star aligner. So these are the things. Yes.
Quick question, what's the advantage of one over the other?
Yes,
I mean,
so
that's the key thing.
Right. So the advantage of this is that it's an order of magnitude time-wise faster. The disadvantage of this star is that it's much bigger, because it creates a whole genome tree and loads it into your memory. It takes a lot more memory than this. So if memory is an issue on your computer or your server, use bowtie. If memory is not an issue, you can load a huge amount of data. I'm talking like 16 gigs of memory. If you have more than that on your computer or your server, then use star.
So for Eris1,
We have.
Okay, cool. So we should be using star.
It's not in my place. You can see the advantages and disadvantages depending on your data. If you're looking for a specific kind of splice variants and stuff, Tophat says that they optimize it, but star is also very good. They're both really good aligners. It's just for RNA-seq specifically, I prefer star. Okay, as Mo was saying, you could use both. Some people would use BWA aligner. For SNP calling, there's one aligner some people prefer. So you can go over these forums. So that's why I put this thing over here, this link. So you can go to these forums. On Seek Answers is a good website in general. If you have doubts about these exact questions, you can go and see what the community. That's exactly what I do. So at a given point, if somebody, because things keep changing, new algorithms and new stuff keeps coming into these aligners. So when I'm not sure what I want to do, I just go to the internet and Google that and then just find the answer and use that. So this is how we found out about that star is better at this stuff and Tophat is better at that stuff. But a word of caution here: you cannot always trust the internet. So try it out yourself before you decide that this is in fact the case. Okay?
Do we get the same result using different
No.
using the same data?
No. They're based on different algorithms. The backend alignment algorithm, by which I mean is the way they have coded their procedure, is very different from both of them, for both of them. So it's very unlikely that you will get the same exact result. You may, in some exceptional cases, it might give the same exact result, but in my experience, whenever I've done alignment using two different algorithms, it's always been different, and which leads to downstream analysis being different as well.
Yeah.
So
yeah, in that situation, then how do you know which is
the better one?
you
Yeah, well,
trial and error, I guess.
Trial and error, exactly. So a lot of bioinformatics, in this kind of stuff, that's why you have so many different aligners. is because one will offer some advantages, and you have to try them out to figure out. And there are papers that have been written just on comparing algorithms. So like for RNA-seq, I can officially say that yes, star works better than Tophat. Okay, but I mean, again, for ChIP-seq, I haven't really compared across the breadth. I don't do so many ChIP-seq. But I mean, if I was to use, I would still just start with star, and then go and search the literature and see which other aligners are there. Most of the time, it's not like when you say better, it's really makes a huge difference. Maybe you'll not find a couple of genes using star, which you find using Tophat, or vice versa.
That's my question. What's
Right.
How much difference is it?
It's not. That's a quantity-wise, it's not that different. So if you suppose you don't find your gene of interest, in that case you can just go and try to do it with another algorithm to see if it pops up using that other separate alignment. But if you end up finding it with both, then that's a confirmation that your hypothesis was correct. Okay?
You don't happen to know which one Ben Whitner uses, do you?
Ben uses Tophat.
Yeah,
I think he may be switching to star. So
okay.
I guess there's also a difference between organism to organism.
Yeah.
Whichever works better.
exactly. Yeah. So for human and mouse, I've used star, and it seems to be doing okay. Okay. So today we'll do Tophat 2 aligner. Again, just because I use star, it still doesn't mean that Tophat is not used. It's a very popular aligner. People use it, and it's a good aligner. There's please do not quote me saying that Tophat is not a good aligner. I don't want Cole coming here and just speaking. Like, what would you just say?
After I would talk with my host.
Yeah. All right. So it's good for splice junction mapping for RNA-seq reads. It's fast. It can align to mammalian-sized genomes, and it uses bow tie aligner in the back end. So you have the option. So for example, when I was working on color space reads, star does not use color space reads. So solid sequence data is color spaced. So the quality, the calling, the way it's done, it's different from Illumina. So in that case, I used Tophat 2 with bowtie 1, because bowtie 2 does not do color space alignments. So Tophat 2 with bowtie 1 was the choice. So again, it varies based on the data, based on the machine, and it has a lot of detailed options for alignment. I can use it for SNP calling, transcript counting, all done afterwards. So all of these steps are done afterwards, but it has a lot of detailed options, and that's what the manual was for when I sent that to you. This is the usage. So we'll go over this slide in a little bit of detail, because this is essentially what you need to know when you're putting the command on the server. When you're running, this is the command you run. So the greater than sign is just a prompt. It's just saying that this is where we start. So Tophat is the actual program name that you run. So I believe if it's Tophat 2, there may be a 2 added to it, but Tophat. Then so the -GTF is specifying what GTF file that you want to use. So you can run it, I believe you can run it without the GTF as well. But if you're doing junction calling or something like that, you need to specify the GTF, because that's the reference that you want. By default, it can also create its own GTF file from your data. Do you know what I'm saying? So it can assemble that and create its own reference, and then use that to further realign. But it's always better to specify a GTF file that you download from UCSC Genome Browser, the table browser, the one that I just showed you. Okay, or and then so this -- output directory is your output folder, basically the folder where you want to store your output files from Tophat into. Okay, by default it'll just store it, I think, in your directory where you're running. So if you don't want that, then do this. This is the next. You give a space. So notice the space after everything. You can see here where there's a space here. And then same thing here between. So when I put things in these, think of these as just like placeholders where your actual file name will go. All right. So this sample output folder directory name goes here. Space. This is where your genome file is located. So your genome file will be your reference FASTA file. the human genome reference. So you give the name to the folder. You don't have to specify the file. I think I'll just search for the within that folder for the reference FASTA file. And then these are your FASTQ files. So this could be, normally they come off the sequencer, they'll have a sample ID _1. So you can take pair. If you have multiple files for the same sample, because that can happen sometimes, you can specify that by putting comma and sample. Say this was sample 1_1, so you can do sample 2_1, specifying that it's the same sample, but you separate them with commas. And then when you do a space, it changes over to the next. Space means that that separator is for the next sample. Okay? So you can align multiple samples at the same time using this. Alright? Yes?
I seem to remember there being like the left paired end and the right.
Yeah, so that would be so this will be the left and then this will be the right.
So it's the same sample for those two, but one is the left and one is the right.
Right.
But if you're doing multiple samples in the same command,
Same
you put a comma.
Yes, but you can also put comma for see, think of it as it's the same sample but split over multiple lanes, so it'll create a different file.
Okay, gotcha.
So then you can separate them by commas. But otherwise it's left and right paired.
Okay. So if you wouldn't do, like, let's say you're running 3 biologic replicates in the same lane.
You could treat them, if you want to just treat them as one sample, you could just do that as well. If you separate by comma, then it'll treat it as one sample.
Okay, I see. Okay. So then,
But you
wanted
can
to
align it
separate
separately.
everything.
Yeah.
You wanted-
If you want a separate output file,
Yeah.
the question is, do you want the same output file, all the reads in one file? Because I've done that. I've taken all the replicates and put them in one. It just outputs everything into one file.
Okay, gotcha.
Alright? So if you want, you can do that.
Thanks.
So just a quick question, it's kind of like one step back. So are you actually creating a new folder, and under that you're creating a new file? Is that what you're doing in that process too?
In this?
Yeah.
Yeah.
So that creates the first command, or the first part of it, sample output folder, that creates the folder, and then...
No, it won't create. The folder has to exist.
Okay, so you have to, okay, that's what I wanted to know.
So yeah. You create that beforehand and then just specify it.
And it just goes in there, got it.
So in terms of the output for your aligner, so this file, accepted hits.bam, is a now, are you familiar with the SAM format and the BAM format? I mentioned it briefly, but okay, we'll go over it in the next class. But think of it as a sequence alignment format where, apart from just your read sequence, it specifies the location where it mapped to. So that's it contains information of where this read mapped to, if it's a multiple mapping or a single unique mapping read. What is the number of bases matched versus mismatched. All of that is stored in your output, right? So the purpose of the aligner is to give you that information. The whole point why you're aligning your FASTQ file is to know where it aligns. And that information about the alignment itself is stored in a file called SAM, so sequence alignment mapping for file. Now, BAM is just a binary version of that file. So in order to save space, we end up converting a SAM file. SAM files can be, big. So if it's an 8 GB file, a BAM would be 2 GB. So it's like 4 times compressed a zip file. Except it's in binary format. And so this is the default output by Tophat. So accepted hits is just the name that it gives to the output. So, and then the next output is the junctions.bed, which contains information including genomic location and supporting evidence about all of the splice junctions discovered by Tophat in the process of aligning the reads. Now you may not use this file, so that might be your primary objective, is to find out all the splice junctions. So based on your requirements, we usually end up using this file more because that's what we're looking for, is the downstream in an RNA-seq. We don't really care about the splice junctions, but they're there in this file in case you want to go through it. And it's in a bed format. Again, in the next lecture we'll be going over SAM tools, bed tools, and going over what their format is and how we can go from SAM to BAM and BAM to SAM, vice versa. We'll also be doing some snip calling in the next lecture in the practical. Okay?
So this is done automatically by Tophat every
Yes.
time BAM has it.
Every time. For any sample.
Can you turn that off? Would that speed it up if you turn it off?
Here, I'm just mentioning this here because it is an option available as part of the Tophat suite. So normally, when I'm running my programs, I end up stopping at the accepted hits.bam and then using that file via a separate process to create reads, and then from those read counts do the differential expression in R, which we'll be covering in 2 lectures from now. So, but that option is available via this. If you have a novel genome that the reference is not available, you can use this to assemble those transcripts and estimate their abundances and all that. So it has all these features and they're quite useful, to be frank. So it accepts aligned RNA-seq reads and assembles these alignments into a parsimonious set of transcripts as well for you. So that Cufflinks then estimates the relative abundance of these transcripts based on how many reads support each one. Again, it's a very specific case of what you may or may not need.
Does it do normalization total number of reads per
Yeah,
second?
so the cuffdiff,
So that's already in there.
cufflinks, and then the program that does that is called cuffdiff. So like for differential expression. So it does the normalization and gives you the differentially expressed genes. And the thing about this, the advantage about using cufflinks and cuffdiff is that they're written by the same person, so or the same group at the very least. So they have a very streamlined way of going from accepted hits.bam to using cufflinks to using cuffdiff. It's just basically 3 lines of code. Go from one to the other to the next, and you're done with your differential expression. The disadvantage is that the output is not, it does not, how do I say this? in the best possible manner, it does not look as promising as you do when you use other softwares like count your reads using separate softwares to count your reads, and as well as to do differential expression using other softwares.
Yeah, I've seen huge differences between the way it's presented through cufflinks and other
Yes.
algorithms. Like
So-
really big differences.
Right. So that's so yes. So I didn't say that you did. So but that's true. But that is true.
I mean, I wasn't doing any analysis of stuff Ben
Right,
did.
right.
He did it in multiple
Yes.
ways.
So that is why a lot of the bioinformatics community has moved to using Tophat as the aligner, which happens to be still its strength. And then downstream analysis is done using some other program to count your transcripts and your reads, how they align to those transcripts. And then using for differential expression, people use multiple programs, and we will be covering that process as we move along. So so going from the next step, once we align it, then after that we'll go over to the next step, which is counting your reads. Once we understand the file formats, so once we understand what SAM tools is, BET tools is, then we go over into counting your reads, and then that's when we start R. So you see, like it's gonna flow straight into R, and you'll then do your differential expression analysis. So that's where we're headed.
Is cufflinks just more stringent or something in its differential expression calls? Like, why does it give you, like, quote unquote, less results or worse results?
It's not less, it's just different. So the call that it makes for a certain gene as differentially expressed may not be coming out to be. It's based on the statistics and the way they're modeling that differential expression is. As you learn, you have to model the data on a distribution, and then based on how that is done and how the comparison is done and how you correct for your multiple testing, all those things combined give you the list of genes that are differentially expressed.
And you're on balance, cufflinks will give you less differentially expressed genes.
Cuff diff, yes. Cufflinks, cuff diff combination, not less, different.
What-
So it's just a statistical engine that's different, that's
Right,
behind
right.
here versus
Well,
the
right,
programs.
right. So the results do not match up. So in R, if you do the differential expression using multiple softwares, you get a lot more overlap between those results versus cufflinks has a less of an overlap in the genes that are called as significant or called as differentially expressed.
Isn't it a huge problem? That sounds like a huge problem.
Yes, and that's why, you know, we go over other methods. Otherwise, I would just basically be like, all right, here are 2 other additional lines. You add those lines, and you're done with RNA-seq. So we don't even have to go into R that much then. You know, other than maybe for ChIP-seq there's a package or something, but you don't need to really go into R that much. But that's the whole reason why we cover that. Okay, so now we'll go over star aligner, the command that that is needed to run this, and this is exactly taken from my pipeline file. So notice that, you know, instead of just using Tophat, you literally just have to go module load star, and then use these commands like 1 and 2, instead of the previous one where you were going module load Tophat and running Tophat. So here. So instead of running this command, you just come and run this one. This is more to generate your index. And I'm putting it here because, well, Tophat also has a similar command to generate your index, but most of the time those are also available in your server. But, you know, star has a different index because it uses a suffix tree as opposed to a Burroughs-Wheeler's transform. So you have to generate this in case it is not there. So check with your bioinformatician, or if you are sure it's not there, just go ahead and generate. So the first line which is omitted here is module load star aligner, and then or module load Tophat, and then you follow this command, which is exactly like this: star -- run mode genome generate. So run mode space genome generate, one word, space -- genome directory. This is where you want to store your. So here I'm using these curly braces to specify instead of the the greater than less than symbol, because this is exactly this is in Python. So the reason I'm using these curly braces is because I literally copy pasted from my script. So that's why this is different. And the Tophat one I wrote, so the syntax is different. But basically, index directory is your directory where you want to store the index. And you say for any index you need a FASTA file, because that is the file that the index will be off. So it will be created off of. So that's why you need this -- genome FASTA files where you specify your genome.fa. And and then if you want to specify the GTF file, the one that you downloaded, you can specify that using this sjgb GTF file space GTF. Okay? So this is just the index generation step. Right? Again, Tophat has also got a similar index generation step where you say Tophat index and give the FASTA file, and it'll create an index for you, and minus genome directory or output directory, and it'll output it there. Now the actual alignment happens using this step. A couple of things are different here in the syntax. So you have to specify a temp directory where Tophat ends up storing its temporary files, which are used as part of the running of the program. So it generates these temporary files. And and I think it deletes it in the end. If you don't want them stored, if you want them stored, you can specify it in the manual. There's a command, but most of the times you don't need them. So -- genome directory tells you where your index is. So this, what you generated here, is the same as this here. Okay? That's where your index is stored. That's what it needs. And then the genome FASTA file is the same file as the one that you use to generate the index, the same file that will come here. The GTF file also stays the same. This is where it knows that these are the transcripts, these are the coding sequence, all that stuff sits here. And then, now the output SAM type. So this is where you can specify whether you want a binary output or you want a SAM, a simple alignment file. So again, we'll cover this, the SAM and BAM, but basically whether you want it in a zip binary format versus you want legible, not English, but readable format is the output. And then sorted by coordinate -- so So here, this is BAM, and then you want to also say if you want it to be sorted by coordinate. So this again we'll cover next time, but BAM files or STAMP files, when they're aligned, they're not sorted. But a lot of programs downstream need these files to be sorted by their coordinates. So by coordinates meaning like chromosome 1 should be before chromosome 2 comes along and stuff like that, right? So all the reads that align to chromosome 1 should be first, and then chromosome 2 should be next, like that. So now, I know I've written these there, but a word of caution here, when you have a big file, this step can take a lot of time. So if you're short of time, just omit this entire output SAM time sorted by coordinate, okay? And then instead, sort it using SAM tools, which we'll cover next time, okay? So this will save you a lot of time. I'm talking days worth of time. This takes a very long time, and I learned this the hard way. I was working on a data set, I couldn't figure out for the life of me why it was taking so long to finish alignments, but it was because of this flag, output SAM type BAM sorted by coordinate. That was taking hours and hours and hours of extra time. Whereas in SAM tools, this step is implemented a lot better, and it takes much less time to sort using SAM tools. Okay? All right? This is just a tip. So, and then here is the prefix for your output file name, where you specify the directory where it creates the file for that you want written out. And finally, you have to specify your read files in FASTQ-R1 and FASTQ-R2. So again, same thing. Here the input is also same way. You have your one file and your two file. It's for paired end. If you don't have paired end, you can just put one file. If you have multiple replicates of that R1 or of a single file, you can separate them by commas. So same exact thing as Tophat. All right? So any questions still here? We'll be doing all of this today. So, all right. Okay, I guess it did take for these 5 slides, it almost took an hour. All right, so then you copy this. This is just a simple command because it outputs the file like this. It's called aligned.sorted by coordinate.out.bam. So if you want to keep this name, which is very generic and it's the same name, and if all your files are being output in the same directory, it'll be replaced next time a new file is aligned. I highly recommend that you use this copy command and copy this to another file that you really want to be your true output file name. So that would be your sample1.bam, right? Because what will happen is next time you align something, it'll give the same directory, it'll give the exact same file name, so it'll be overwritten. You don't want that. Okay, and then finally, because this occupies space, so you just remove this to free up the memory. Okay, that's it. So we'll.
One quick question.
Yes.
Sorry, when it's creating the temporary directory,
Yeah.
does it take up a lot of memory? So it's using RAM, right? So
Yeah.
Yeah.
So this is temporary files that it writes during the process of its, so it needs to store some variables, store some files, stuff like that. It does take up some memory. The memory that is taken up is not so much of the temporary directory. It's more for the RAM that is for the suffix tree that is loaded
Sure.
into the memory,
Yeah.
which is order of like GBs and GBs.
I mean, depending on the size.
Of the genome, yeah. If you're fly genome versus a human genome, yeah,
It's
obviously.
going to be a huge difference. Okay.
Okay, so we'll take a 5-minute break and let's reconvene and start our practical right away. 5 minutes break.
So does the index already exist for star, like on our server? Has somebody already done that?
Yes.
Okay.